present our model and try to synthesize what we have learned during the years working with this animal. For those that are not familiar with the cotton rat, this is a new world rodent that we have been working with many respiratory pathogens, influenza, rhinovirus, adenovirus, metapneumovirus, and for sure you certainly know the cotton rat for all the studies and translational studies and value that it gives to the therapy with antibodies for RSV. So in terms of defining clear endpoints of enhanced RSV disease, what I wanted to show you first is different aspects of disease that you can manipulate or generate in this model. And for that, I will start with the slide just defining simple models of disease, starting with what is primary infection, that as you infect cotton rats, it generates a high level of replication in the lung with a peak of around day 4, day 5, and start decreasing by day 10. So these are naive animals that have been infected, so they are really very good for replicating the virus in the nose and in the lung, and still are able to produce good level of lung disease that is basically confined to peribronchiolitis and bronchitis. So then we start thinking, well, what happened with secondary infection, so animals that has been seen the virus before, and as you can see, these animals are fully protected against replication in the nose and in the lung. And still, what is interesting is that they have similar levels of pathology as seen in primary infection. Even the virus is not replicating in the lung. We have described the presence of abortive replication in these animals in the lung, indicating that probably some component of the virus are produced, and for some reason, they can be detected by the immune system and also generating disease. So different backgrounds of immunity cause different levels of pathology. So for sure, we have been doing experiments with FRSV, formalin inactivated RSV preparation that was the original use in the trials, is the lot 100. And what you find is that there is not complete protection, as you will see with primary infection, but still you have a reduction in viral replication in the animals. What is clearly seen in these animals is that you could see enhancement of pathology in the lung, and mostly this is a characteristic appearance of one of the components of pathology, that is alveolitis, or immunocells that are coming into the alveolar space. So, with all this, we came to some kind of parallels of vaccine-enhanced disease that has been shown in humans, like alveolitis, so primary PMLs, and lymphocytes that infiltrate in the lung as characteristic pathology of FRSV. Also, we have seen, in this case, insufficient induction of neutralizing antibody that corresponds to the decrease in the viral replication, but still you cannot produce complete immunity. What we have shown and in recent years with the increased reagent for cotton rat is that there is exacerbated immunoresponse characteristic for cytokine chemokine storm. So as I mentioned to you, different levels of immunity will cause different levels of disease, and we have change different parameters like immunosuppressing animals, or also we have work with different age of cotton rats, and we see variations in this pattern of disease. So indicating that the immune system and the immunocompetence of the animal will basically going to affect the final parameters that you will see in disease. And you will have different changes in the induction of neutralizing antibodies, and also in the case of immunosuppressed animals, you will see extended level of replication. So, in terms of defining components of vaccine enhancement that can be used, we decide to go forward to models that will mostly reflect landscape of vaccines that are now being developed. And this is the case of, by example, maternal immunization. So we start asking questions about, could babies born from mothers vaccinated with the prototype vaccine X develop signs of enhanced disease upon RSV infection? So basically, if you vaccinate the mother with a vaccine that is bad, do you think that you can transfer bad immunity to the babies? And these are questions that we are trying to ask in the model now. At the same time, we will ask, would baby vaccinated with the prototype vaccine X early in life, while still in the presence of passive transfer maternal immunity, develop signs of enhanced disease upon RSV infection? So these are questions that has not been answered in models and need to be taken in consideration for several different populations of vaccines. So I will leave this here.
Thank you very much. All right, so I wanted to talk a little bit about RSV vaccine-enhanced disease. So if you could adjust the size again. So we really think RSV vaccine-enhanced disease that was caused by the formalin-inactivated RSV vaccination was the result of a perfect storm of events. The first was that the formalin-activated RSV vaccine elicited a poorly neutralizing antibody response. It also failed to elicit a CD8 memory T-cell response. And it elicited a robust memory CD4 T-cell response because the vaccination was essentially a bolus of dead antigen that was efficiently presented in the class 2 pathway. And because of this, when the vaccinated individuals saw a live RSV infection for the first time, they mounted a very strong and robust memory CD4 T-cell response in the face of a large amount of viral antigen replication, but they lacked that memory CD8 T-cell response to limit viral replication, and they lacked a neutralizing antibody response to limit viral spread. So they got an overexuberant CD4 T-cell response that was really the main driver of pathology. We also showed last year that eosinophil deficient animals do not show any differences in RSV vaccine-enhanced disease, despite their implications over the last 50 years as a major contributor to the pathogenesis. We actually believe that the eosinophils were trying to help repair some of the damage in the lungs that had occurred by the robust CD4 T-cell response. They're a very good marker of strong Th2 immune responses, but in the case of RSV vaccine-enhanced disease, we don't believe they contributed directly to pathogenesis. And we were also able to show that both CD4, Th1, and Th2 subsets contributed to various aspects of RSV vaccine-enhanced disease. The Th2s drove a tremendous amount of the pulmonary inflammation and changes in the airways and contributed to airway resistance, as well as mucus production. The Th1s drove a lot of the systemic disease impacts, including weight loss, but they also contributed to airway obstruction, so they may contribute to some of the epithelial cell sloughing that occurs. So of course, everyone wants to know markers of RSV vaccine-enhanced disease. I think the primary marker really is the induction of a poorly neutralizing antibody response. This really sets up an environment that is conducive to the generation of RSV vaccine-enhanced disease. And I think most current vaccination efforts would not create this environment. And so I think this is really less of an issue. But basically, if your vaccination elicits a poorly neutralizing antibody response, that's going to be problematic. We also know that robust Th2 cytokine responses are a major contributor to the pulmonary pathology that occurred in RSV vaccine-enhanced disease. So if that is picked up in a vaccine trial, that's a poor indicator as well. Eosinophils have often been used as a marker, but again, we think they're just simply a marker of a very robust Th2 response. And so they can still be used as a marker of that Th2 response. But we still are lacking significant biomarkers for RSV vaccine-enhanced disease. I think, again, the best one currently is the neutralizing antibody response. And then what we really don't know is, again, in the vaccine trial, only 2 of the children succumbed to the RSV infection out of the hundreds that were vaccinated. And we have very little information on those children. It's possible that there's a genetic influence as well that we're unaware of that could be a contributor to vaccine-enhanced disease. And that's something we really need to explore a little bit more, particularly in animal models. And then finally, I want to talk about some of the causes of severe disease in just a normal, healthy child. So we often talk about balanced T-cell responses, but I think in part we want to talk about balanced humoral versus cell-mediated immune responses, because I think that's really where you want the balance. And so we know that in the absence of a strong neutralizing antibody response, that robust RSV-specific T-cell responses can lead to immunopathology. Both Th1 and Th2 cells have the capacity to create immunopathology in RSV infection. And that's an important point because I think a lot of people think that Th1s are always protective in an RSV infection, but there can be a number of cases where overly robust Th1 responses can lead to immunopathology. And finally, I think we really need to get a better handle on the number and the functional phenotype of memory CD4 and CD8 T cells that contribute to optimal protective immunity. We have a really good handle on the amount of antibody responses that we need to protect against disease in particular. But the other side of the coin is where we're really lacking in our assays and our ability to measure cell-mediated immunity. And I think in order to elicit optimal protection following vaccination, we need both humoral and cell-mediated immunity. And we need to get a better and do a better job of measuring cell-mediated immunity following these vaccinations. And with that, I'll stop.
Good morning. So when I think about this, I want to divide things into what type of antigen you have, and then go into host factors that may contribute. So you look at the antigen, you look at the host, and in the host you look at the pattern of immune response and also the cause of disease or the pathology. And so first, thinking about the antigen that was used, this Lot 100 vaccine was a formalin-activated product that was made by baking the virus at 37 degrees for 72 hours in the presence of a low dilution of formalin, 0.04%. And so because of the reagents that we've generated recently and some of the things that you saw Jose present and then Jason showed some data on how to look at the kind of antigen on the surface of a virus, we've been able to show recently that as virus dies in heat at 37 degrees over time, that one of the factors in that loss of infectivity is the loss of the functional F molecule on the surface of virions, and so it flips into the post-fusion state. And what that looks like using these dot blots is that even though you preserve F on the surface, shown by the modivizumab site 2 that's on the shared surface of pre- and post-fusion F, you lose pre-fusion F based on binding of these either the quaternary trimer-specific epitope that Jason showed, AM14, or the site 0 specific epitopes shown here. So as virus dies in heat, it loses pre-fusion F, and so you're changing the topology or the antigenic capacity of the virus. And just, whoops, I lost something there. Oh, there it is. So I'm just showing you again this pre-fusion F structure, and there are now hundreds and hundreds of RSV-specific antibodies that you can show cover the entire surface of F. So just showing on one protomer, there's antibodies that cover all these different antigenic regions of the molecule, and you can so generally divide those into 6 regions, and you can then rank them by their neutralizing potency, somewhat from top to bottom, but mostly based on whether they're present on the pre-fusion functional form of the protein, whether they're on the shared surfaces of the pre- and the post-fusion F, or whether they're mostly represented on the post-fusion form of the F molecule. So the most potent epitopes, or neutralization-sensitive epitopes, are at the apex, and in this region here, defined by the MPE8 antibody, or in this region here, which is somewhat of a new antigenic region that you'll be seeing antibodies described for. And these pre-fusion specific epitopes are the most potent, and that's where you're going to get your high neutralizing activity, either in primary induction of an immune response or as a boost to a pre-existing immune response, because even these antibodies, the site 2 and site 4 in particular, that have neutralizing capacity, they see the epitopes in the pre-fusion context differently than they see the epitopes in the post-fusion context. And so just reviewing again, this is Steve already has shown this, the 2 main host responses that have been associated with the enhanced illness syndrome are this idea that you have poor neutralization activity that then presumably contributes to this immune complex deposition that was demonstrated by Fernando in an earlier paper from lungs from the children who died in that 1966 trial. And so poor NEUT activity with immune complex deposition may be one facet of the vaccine-enhanced illness. And then this pattern of response that includes Th2-like cytokines that Steve just described is another marker or facet that's been described in animal models, but we don't really know how those animal models are going to translate into humans, especially young, naive, developing humans. And so these are just some things to think about during the discussion. I'm going to stop there and turn it over to Peter.
Sum up a few thoughts about pathogenesis, and just to focus your minds on the importance of, if you can come up, of sequence and timing and of age. So let's think about all the factors that might together come to combine to cause severe disease. So clearly the virus is one, and we don't know whether the sequence variation in virus might be contributing to disease, but my suspicion is that maybe there's combinations of particular viruses with particular hosts that cause severe pathogenicity. Remember that many children only have mild disease. Many of them may not even be symptomatic. So I think we need to understand that better. So what's the effect of viral load and how can we measure it? I think that's quite difficult. We're going to hear about the host factors from Stephen Kleberger later, which I think is going to be really, really great. And we've been thinking a bit about the environmental factors, the pre-existing microbiome, and what happens when the virus collides with the host and causes all this damage, the role of bacteria, the role of mediators. So putting this together in a sequence, in a timing sequence, again, I want to emphasize the vital importance of time as a determinant of what's happening. So viral entry could be blocked by IgA and other local mucosal factors, particularly the sort of mucosal tone that may be determined by infection history and by environmental stimuli. In particular, maybe the microbiome could determine this mucosal tone. So once the virus has got through an established infection, then viral replication both results in and is controlled by the host immune response. And we haven't heard much about NK cells, but they may also be very important. And T cells may both be pathogenic, but also may assist viral clearance in particular. And I think those very early experiments that I described right at the beginning of this morning's session, where we injected CD8 T cells into the tail veins of mice and they caused enhanced and severe disease, maybe we were creating a situation in which the wrong cells were there at the wrong time, and that was really what was causing that very severe neutrophilic inflammation that we saw in those very early studies. And we also haven't thought much about resolution and convalescence. So how does this acute viral hit resolve? What are the later consequences in terms of maybe, lung scarring or changes in the basement membrane or recurrent wheeze in later childhood? So I think, timing is of the essence, as is age. Now, I'm increasingly thinking about age after all these years of working with RSV. So clearly, we started off thinking about RSV as being very much a childhood disorder. And I think many studies, particularly the elegant studies from Rochester, have shown the importance of RSV in old age and emphasized that a lot of the mortality attributable to RSV disease in statistical models is actually in elderly people rather than in very young babies. So there's birth. There's also, I think we need to think about infancy and young children. So what role do children play in transmitting disease? And would they be a suitable target for vaccination? And then in adulthood, we need to think about pregnant women and vaccination of that particular group because of the possibility that they could protect children by passive antibody, and so into later adulthood. So I think we need to think across the spectrum of age in order to really understand the disease in all its different manifestations. And what may apply in infancy in terms of the types of immunity which are important or which can be induced may be very different from the types of immunity that can be induced in later life. So I think we're thinking about both the timing of the infection itself and also the changes that occur as life develops are very important to understanding RSV disease in its entirety. So I hope during our presentations you've been thinking of some good questions, because we very much want to make this a participatory discussion, because there are so many of you in the audience who are real experts. So I would really invite you to come up to the microphones and to contribute your discussion, and I will go and sit down. Thank you.
Hi, very nice presentations. I think, so the title of the panel is Enhanced Disease, and I think this could imply either FIRSV enhanced disease upon infection or severe disease with natural infection. So I was wondering if the panelists want to give their thoughts on the similarity and difference between those two. Thanks.
Barney?
Okay, well, I think largely the basis of disease in these very small infants is obstruction of airways. And so the question is, how do you get to the obstructed airway? There's mucus production, which could be provoked by the Th2 type background. There's sloughing of cells, there's fibrin, there's edema, et cetera. And so I think what happened in the vaccine-enhanced disease is just an exacerbation of what can happen in some small percentage of normal infants, and that is with their priming and pre-existing immune response, that they had an ineffective immune response that led to more of these factors that ended up clogging up the airways, causing obstruction in this case, 80% of children instead of about 2 to 3% of children. And so I really think the basis for ultimately the bottom line disease is obstruction. And the question is, do you have an effective immune response that clears and prevents this obstruction, or do you have a good or an ineffective response that exacerbates it?
Stephen, do you want to comment on that?
Yeah, I agree. I think, in infants in particular, the younger the child, the less effective the CD8 T cells are in an acute infection, and I think that's a major contributor to why disease is much more severe in a very young infant versus one that gets their initial RSV infection at 1.5, 2 years of age, where their cell-mediated immunity is a little more effective in knocking down viral titers. So I don't think you get much of the CD4-driven pathology in the face of that. So I think that's a really important factor that needs to be considered.
I mean, I guess to me, a thread that's come up again and again. So Chris Chu was asked, why aren't we sampling the local bronchial lymph nodes? Well, trying to get at the site that really matters is a major challenge, especially in vulnerable babies, where increasingly we're being non-interventionist. Yes, so we've got lots of questions. So.
Hi, Jose Malero from Madrid. I would like to add a little bit more information about the antibody responses that we were, particularly Barney was mentioning, in consideration to the possibility of contributing also to pathology. And it was proposed by, or in the studies by Fernando was seen. We recently evaluated the antibody, the protected immunity in mice in collaboration with Geraldine Taylor, using pre-fusion and post-fusion F proteins. And what we observe is that when we use a high dose of the antigen, both pre and post-fusion were protective, and the best neutralizing antibody responses were obtained with the pre-fusion protein. But at low doses, where we found some mice that were protected, others were not protected. When we look at the antibody responses in those mice, the titer of antibodies, specifically for the pre-fusion, by ELISA, were as high as those that were protected, and yet the neutralizing activity of those sera was essentially none. So you can have antibody responses that you can distinguish by ELISA and neutralization. So you can have a good neutralizing antibody, a good ELISA antibody titer, and yet essentially no neutralizing antibody response with the pre-fusion protein. So I think this is just a sort of call of attention to those that are developing vaccines, that you have to look carefully to the type of antibodies that you are inducing, because those antibodies might, as Fernando was suggesting in his studies, that those antibodies may not even protect, but could contribute to the pathology.
Can I just ask whether anyone would like to say that they've seen infection-enhancing antibody? So, I mean, there's been discussion about this for years. I think Eric Simos. Eric, are you in the audience now? So I think in the bonnet monkeys, Eric demonstrated passive transfer of antibody actually enhanced infection. Do you recollect this, Bonnie?
Eric would have to say, I mean, there are some settings in cell culture that you can enhance slightly with certain antibodies infection, but I have never seen anything like this in vivo. And even in diseases like dengue and others that are monocyte tropic, where FC receptors are critical for entry and where that kind of antibody-enhanced infection can occur, it's very difficult to show this phenomena in vivo, even for dengue.
Okay, we have lots of...
Pedro Piedra. Steve, I like very much your overview, but it also suggests that there are really a variety of different phenotypes that can give you the same type of pathology. And I think that's very true also with RSV bronchiolitis, where there may be a variety of different phenotypes that get you to severe disease. And so one of the questions that I would pose to y'all, and Jose could test in the cotton rat, and that is, if you set up your environment for enhanced vaccine disease, would simply providing neutralizing antibodies prior to the time of challenge abort any evidence of enhanced disease?
Yeah. That is a really good question, and I think that need to be done. Yeah.
Yep. Actually, we have that experiment designed and doing it with various F monoclonals. So we'll be able to answer that for you.
Maybe we can go to the back to...
Can I just
Yeah,
add
sorry.
one more point to that? I think what you're saying is in part related to the contribution of viral antigen versus the contribution of the immune response. And so it's pretty clear that if you don't get virus replication after one of these immunization events, you don't generate much pathology at all. And so if you reach a threshold where you can prevent virus replication, it's very hard to demonstrate pathology in these murine systems. And so one of the things I think you have to look at in the murine systems evaluating for the enhanced pathology phenotype is, did virus replicate at all? And if virus didn't replicate at all, it's going to be very hard to see the immune response patterns that might be causal.
Well, this is different in the cotton rat. In the cotton rat, we see complete protection of animals with good titers of neutralizing antibodies, and we see still pathology in the animals. And
But
this
do you see illness?
Hmm?
Do you see illness?
Pathology in the lung, no. We don't measure illness, but we have markers of interferon production early that are shut down after infection. We have demonstrated that there are abortive replication, and we have several cytokines that are expressed in the lung. And this has been done over and over, and I showed mostly the three different parameters or setups of infection that we have shown. And this is the most important thing that I think that need to be taken in consideration, what prime animals can add to this idea of enhancement of enhanced pathology, because a lot of kids that has been hospitalized or are hospitalized are probably secondary infected kids.
I seem to remember a study that I think Greg Prince published maybe in the early 90s, late 80s, where he varied the interval between priming and challenge, and that there were different time zones after priming that led to either enhanced disease or no protection.
You mean prime
So-
FRSV?
Yes, FIRSV. So there was an initial window where there was solid protection, then a period during which disease enhancement occurred, and then if the animals were left for a very long period, then the effect again was much diminished. The enhancement was much diminished. But
So you mean basically the time will decrease disease?
yeah,
I don't have that.
okay. I think we need. So there's lots from the audience. Larry.
Larry Anderson, Atlanta, Georgia. The question is going from animal models to being comfortable giving a subunit vaccine to the RSV naive child, and thoughts about that, and maybe in two ways. One is the virus vectored vaccines with the parainfluenza virus vaccine given to young children, expressing F, didn't see enhanced disease, would a different type of vector, adenovirus or others, raise or lessen concerns? And then obviously a vaccine where there's no replication. Strategies to get to that point of actually giving to the RSV naive child.
I guess a recurring question is, what have basic immunologists ever done for vaccinology? I see Alton Power at the microphone back here. Alton, with your experience of
Thanks,
vaccines.
Peter. Alton Power, Queen's University, Belfast. In my previous existence in the pharma, we worked with a small molecule, BBG2NA, which is central conserved domain of the RSVG protein. And just a comment and a couple of questions. Comment relates to what Jorge was saying about the presence or the safety of vaccines in the presence of maternal antibodies. We looked at that in mouse model, where we infected the mice, mated them, and then vaccinated the pups within 7 days. Waited until the maternal antibodies had disappeared and then challenged them. And we found no evidence of any enhanced pathology at all in the presence of vaccination in the presence of maternal antibodies. So just a comment
So you
on that.
vaccinated with RSV the pups?
No, this was with live RSV, vaccinated with BBG2NA in the presence of antibodies derived from live RSV infected dams. So no problem there under those circumstances. Comments relate to, first of all, in FIRSV in mice, at least in our hands, and I think other people have seen the same, it clearly induces enhanced pathology type symptoms, but it's also fully protective. How do you relate to that, or what's the panel have any comment on that? In terms of the dissociation between enhanced pathology and actually protection. And this probably relates to the idea of what exactly is neutralization or neutralizing antibodies in vitro. How does that refer to what's happening in vivo? And the second comment relates to some of the work that was discussed earlier, and this is related to the microbiota. And it's something we probably haven't discussed a lot about, is that in the 2 kids that died in the FIRSV study, one was infected with E. coli and the other was infected with Klebsiella. Do we need to include that in view of the enhanced disease that we seem to be associating with the presence of bacteria in the enhanced pathology models that we're actually looking at?
I think Alton's raised lots of questions. We still have quite a lot of people who are wanting to contribute in very little time. Anyone want to make a very brief response?
So in our hands, I think it depends on how much virus you challenge the FIRSV immunized mice, because we get replication even in the face of the immune response that develops. And so I think the variation you see may be due to the challenge dose. The other thing I do want to state about the FIRSV vaccine-enhanced disease, because the concept that antibodies mediate that disease and immune complexes, our data suggests that if you deplete the CD4 T cells, you get nothing as far as vaccine-enhanced disease, despite the fact that they have those non-neutralizing antibodies that could have formed immune complexes in the lung. I think the immune complex formation could be a contributor, but I don't think it can be the trigger. And so I think you need the T cell response to be the trigger, and I think that's an important distinction that needs to be.
Okay. So Jerry has been waiting patiently.
Jerry Mastecki, University of Alabama at Birmingham. I was glad to hear that IgA may actually prevent the infection with RSV, as you told us. The only problem is that children don't make whole of IgA, so that immunized children at that age, 6, 7 months, 1 year, will not be too terribly productive with respect to the IgA production. They just don't make it. Second comment, your immune complex picture reminded me of a wonderful paper published some 25 years ago by the best mucosal immunologist, Per Brandsegg, who unfortunately died 2 weeks ago, where he showed very clearly that if you have an immune complex formed in the mucosa of IgG with matching antigen, it will, of course, bring in activation of complement, influx of polymorphonuclear leukocytes, and damage of the tissue. That would be fine, but it also enhances influx or uptake of bystander antigens of any origin. So actually the immune complex formation by complement activating antibodies leads to tissue damage and much enhanced uptake of anything you can imagine, endotoxins, bacterial products, and therefore worsening of the disease. So I think this is something which we should probably remember.
Well, we'll look forward to your lecture later today. It'll be very, very interesting.
I'm not talking about this.
Okay, thank you. Dave.
So we've had a lot of discussion about the presence or absence of antibodies prior to infection. And I'll take up a point that Barney brought up. There's also memory B cells after immunization or infection. And if you have a higher frequency of cells that react to the post-fusion form of F, that's going to inhibit the production of antibodies to the pre-fusion. So you might have a memory B cell competition that goes on that prevents the kind of response that you want to get. Now, this is something that we see in influenza, and actually an emerging story that revaccination may be detrimental in the sense that you start eliciting responses to the wrong part of the hemagglutinin. So I wonder if the same thing goes on in RSV in certain circumstances.
Very, very interesting point. We're really running out of time. Marty, you've been waiting.
Sure. Marty Moore, Emory University. I'm wondering if immune complexes can be reconstituted in vitro with polyclonal and new monoclonal antibodies, if this might be an additional model that could be developed. Just thinking about how these models can de-risk non-replicating vaccines.
It's a very interesting thought. Has anyone?
Maybe, maybe Fernando should answer that question.
Yeah. Good
That's
idea.
a very interesting idea. That's all I can say, I guess.
But, I think the immune complex formation requires the right balance of antibody and antigen. You have to get to that perfect place where the immune complex can really deposit and create the complement activation, and then the immune response to that, either neutrophils or CD4 T cells. And in the models, it's a little bit complicated because at least in the mouse, almost all the virus is replicating in the type 1 pneumocyte, not in the bronchiolar epithelial cell. And so you don't really, you can't fully see what's going to happen to the small airway in some of the animal models.
One thing I thought was important is that if you look laterally, enhanced disease repeats itself in many diseases. I mean, atypical measles is very much, that's how I got to enhanced disease. I was a measles guy. And then dengue hemorrhagic fever is very similar. As Barney pointed out, finding enhancement of infection or infectivity in dengue is extraordinarily difficult, but finding immune complexes and cross-reactive T cells is not. And there's a lot of work that suggests that we even done work in pandemic influenza showing the same thing. So we can derive lessons from other pathogens that are similar. The other thing is that the kids that died, that Steve was pointing out, one had a severe case of croup in the first year of life, and the other had bronchiolitis. So this was the second episode of bronchiolitis of this child. So they may have been a bit special. Greg Prince shared with us many years ago slides from those kids and the autopsy reports. We have a poster the last day showing some of the information on them downstairs.
Okay, thank you. So just one very last comment.
Vahid from Tehran University. My question is about focusing on another molecules beside the T helper 1, T helper 2, or cytokines, because there are lots of molecules like, for example, in my experience, opioid peptides or cannabinoid peptides that have crosstalk with cytokines. And so there is, if you have some balance between this kind of peptides and cytokines, you have also T helper or T helper 1 response or T helper 2 responses. And in my experience, when we added nalmefene as an antagonist of opioid receptors in FIRSV vaccines, after a viral challenge, we saw some depletion of immune responses to TLPR2. It means that it can shift from TLPR2 to TLPR1. I think maybe we have to also focus on other molecules and peptides in severe diseases like RSV.
Thank you. Thank you very much indeed. Thank you. So I think we need to draw the discussion to a close. So I'd just like to thank the panelists and all of you for your discussion. Thank you very much indeed.